Hello, this is Gina Piscatelli with a detailed lecture on the function of DNA in protein synthesis. You have a reading assignment in Chapter 2 and 3, Sections 11 in both chapters, and your last 3 study guide questions, 20 through 22. So let's just review the functions of DNA and RNA before we begin. DNA is the genetic material that we inherit, and every time a cell divides, it divides into 2 cells that receive a complete copy of our genome, all the DNA. All of our DNA, consisting of 46 chromosomes, contains specific genes, and these genes code for polypeptides or proteins. The DNA is located in the nucleus and it does not leave the nucleus. But protein synthesis happens at the ribosomes, which are located in the cytoplasm. Therefore, a copy of each gene must be made and sent out to the ribosome to be used to make a protein. That's where RNA comes in. So RNA is only made, it's a copy of a gene, it's only made when a protein is going to be synthesized that's coded for by that gene. So RNA is initially located in the nucleus. It leaves the nucleus through nuclear pores and goes out to the cytoplasm where ribosomes are located. And ribosomes read this RNA copy of the gene and assemble the appropriate amino acids depending upon what the genetic code dictates. So as an overview, we start with DNA that's double-stranded. Each strand of the double helix is complementary to the other strand. Shown on this slide is a sequence on one strand that reads TAC, CGC, CCT, etc. On the complementary strand, or the other side of the helix, you would have ATG, because that's complementary, and then GCG, GGA. That's what we start off with. If we assume that this entire sequence of DNA is a gene, we need to make a copy of it, our cells need to make a copy of it, in the nucleus, and the copy needs to be RNA. So the process that takes place in order to make a copy is called transcription. So transcription takes place in the nucleus, and a copy of the DNA is made, but the copy is complementary. So the RNA copy is not exactly identical, but is complementary to the original gene sequence. So where we had TAC at the beginning of the DNA, the beginning of the RNA will read A, and then U instead of T. So A is normally complementary to T, but we use uracil instead in RNA, our cells do, and then G. And that continues, that process of transcription continues in the nucleus until we have what is called a primary transcript. That primary transcript is altered somewhat, not shown on this slide, before it leaves the nucleus. When it leaves the nucleus, it goes to ribosomes where translation takes place. And it's the ribosomes that read each codon, or 3 nucleotides at a time. A codon is 3 nucleotides. The ribosome reads each codon at a time and assembles the appropriate amino acid. So we're going to go through the steps more specifically now, starting with the process of transcription. It occurs in the nucleus, and there's an enzyme that binds to the region of the DNA to make the RNA copy. The name of the enzyme is RNA polymerase, and it binds to a region of the DNA called a promoter. And the promoter region has a certain sequence of bases that we don't really have to know, but the RNA polymerase moves along the DNA until it receives, or it reads, a start sequence. And once it reads a start sequence, it begins making an RNA copy that's complementary to one side of the DNA. So RNA polymerase is responsible for synthesizing the primary transcript. Beginning at the start sequence on the DNA template, it moves along in this direction, the arrow is shown right here, and assembles the primary transcript by attaching it to the DNA template. So temporarily, you have a DNA strand bound to an RNA strand. This won't last though. Eventually the RNA will fall off of the DNA and the DNA strand will rebind to its original complementary partner. So the RNA polymerase continues synthesizing the primary strand until it reaches a stop sequence. And as it synthesizes, it puts uracil where you would think thymine would go. So uracil is complementary to adenine in RNA. So overall, transcription is performed by RNA polymerase, which reads 3 nucleotides at a time. 3 nucleotides is called a codon. Each codon codes for one particular amino acid. The next step that happens is called RNA processing, and this happens in the nucleus. The major process that we're going to talk about here is called splicing. There are a few other things that happen, but splicing is what's relevant for us. The DNA, the gene, contains, the original gene, contains some sequences that are not required to make the protein. These sequences might be regulatory sequences or even junk DNA that we've inherited through evolution, but they need to be cut out. So these sequences that are removed are called introns. The introns are spliced out, and they're shown in dark purple in this diagram. After the introns are spliced out, what's remaining are called exons. So A, B, C, D, and E, and F, those are all exons, and they are joined together. Now we have what is called messenger RNA, and I like to think of it as mature RNA. It's just easy to remember. So first the primary transcript is made. That is processed by splicing out introns to make messenger RNA. Now, one thing that's interesting about our genome is that there are regions of the DNA that can actually code for 2 or more different proteins, which seems odd because the definition of a gene way back when I was in school was a region of the DNA that codes for a protein. Well, because of splicing, the mature RNA could have different sequences depending on what's left in and what's taken out. That means that sometimes exons are also spliced. Now this is called alternative splicing versus just regular splicing. Okay, so for example, with this primary transcript, the same one we were looking at before, all of the introns are taken out. An intron comes from intervening sequences. Exon comes from the fact that these sequences are expressed. So exon, the sequence will be expressed. Regardless, the introns are all taken out. And now the exons can be spliced together. Now in our first example, we just spliced all the exons together. And so we had 6 different codons. And technically we would have 6 different amino acids. This picture's not really that accurate. I guess it depends on if this is a codon or not, if A is a codon. So you get a very long protein compared to the idea of not including all of the exons. And this can happen particularly in a cell that can make 2 different enzymes that are similar but not exactly the same. So they're both proteins, but they have slightly different amino acid sequences. So for example, in this mature RNA, the exon A, the exon C, and the exon F was spliced out, and the mature RNA is shorter, so the protein will be shorter. That's called alternative splicing. So as you can see here, you could get quite a different number of combinations of exons, and it's possible that a cell could make many different proteins from the original DNA and primary transcript that was formed complementary to it. But after alternative splicing, the mature RNA is going to leave the nucleus through nuclear pores. And the third step, the last step, is what happens to that mature RNA once it reaches ribosomes. The process is called translation. And during translation, a nucleic acid sequence, RNA, is read by the ribosome. This nucleic acid sequence is read and translated into a different language, the language of amino acids or proteins. So this is a picture of a ribosome moving along the messenger RNA. 2 subunits. Here's one subunit, here's the other subunit. And these are what we call transfer RNAs that bring the amino acids to the ribosome to assemble the protein. And the protein that's being assembled is kind of shown, it's a little short and very dark. It's shown right there. That's a terrible arrow. So the protein that's being assembled is right there. And it'll get longer and longer as the ribosome moves in this direction. So there's different types of RNA involved with this last process, translation. There's messenger RNA, which carries the code from the DNA for protein's amino acid sequence, and it temporarily binds to the ribosome as the ribosome reads it. Now within the ribosome, there's also RNA that makes up part of its structure. There's also protein in ribosomes. And this ribosomal RNA temporarily stabilizes the messenger RNA just to keep it on the ribosome. And then the last type of RNA is the transfer RNA, or tRNA, that brings the amino acids to the ribosome. And the transfer RNA has what is called an anticodon that's complementary to an mRNA codon. So the original messenger RNA had codons. We call them codons. They're just 3 nucleotide bases. And whatever transfer RNA is complementary to that will bring the appropriate amino acid. So I'm going to show you an example here. So for example, here is the messenger RNA codon AAA. What's complementary to AAA is UUU. That is on the transfer RNA and called the anticodon. Every time a transfer RNA has UUU attached to it, and all of this, by the way, is also nucleic acid. It's just that those sequences of bases are not important. But back to what I was saying. Every time a transfer RNA has UUU, it always puts a lysine right there. So that means every time the start codon AAA is read by the ribosome, a lysine will be put in that spot. If I go over to the left and I see what was done before, we see that the messenger RNA was UUC and the complementary anticodon is AAG. Every time a transfer RNA has AAG, a phenylalanine is put on the end of it. And that's how we know what the genetic code is. Somebody has figured out what the codon codes for. Like AAA codes for lysine, all because of this intermediary transfer RNA doing its job correctly. So translation begins with the ribosome binding to the messenger RNA. And then the ribosome scans the messenger RNA for a start codon. And the start codon is AUG, which codes for methionine. So that will typically be the first amino acid in any protein. Now, the ribosome is going to continue reading the messenger RNA, and it moves along the messenger RNA to do that. This movement is called translocation. I put it in 2 places. So translocation is the process. The verb, I guess, would be translocates. So the ribosome moves 3 nucleotides at a time to read each codon. And the only transfer RNA that's going to fit into the ribosome at that particular spot, or that codon, is the one that's complementary to it. So for example, this is lysine right here. And we already talked about lysine. But the one for methionine won't fit here. The one for phenylalanine won't fit here. No other transfer RNA with a different amino acid will work there. And that's what the ribosome does. In addition, what the ribosome does is it catalyzes the formation of peptide bonds. So when methionine, which is green, first showed up, it was kind of hanging out by itself. And then phenylalanine showed up. The ribosome actually helped form or facilitate the formation of the peptide bond between those 2 amino acids. Eventually there will be another peptide bond formed between the blue ball and lysine. Okay. Now the transfer RNA that has dropped off its amino acid, like this one I just circled, it can go get another amino acid and be reused. So that's a benefit. So translation continues as the ribosome translocates along the messenger RNA. But eventually, we need to stop making protein. The protein's long enough. And the way the ribosome knows to stop is one, I guess, running out of messenger RNA. But primarily, it's due to the fact that it reaches what's called a stop codon. And this stop codon does not have a transfer RNA that's complementary to it. So complementary to U would be A, complementary to A would be U, C. So you won't see a transfer RNA with that sequence, and no amino acid will be put there. Eventually, all this machinery falls apart. I don't want to say disintegrates because it stays, every component stays intact, but the messenger RNA will fall off of the ribosome. All the transfer RNAs have left, and what's going to remain is the peptide bonded amino acids. So you'll get a peptide bond. The messenger RNA is intact, but there are enzymes that can break it down so that those nucleotides can be used again to make a new messenger RNA, or maybe for energy, but usually to make a new messenger RNA. So if we look at a summary of this whole process, first we had DNA, which I don't have up here. Okay, so we had DNA, and of course that is in the language of nucleic acids or nucleotides. mRNA is formed complementary to the DNA, and every 3 nucleotides are called codons, or is called a codon. Those codons are read by a ribosome, which facilitates the formation of a polypeptide that is coded for by AUG, the codons, GCG, GGA, etc. However, transfer RNA is needed because it has to bring the amino acid to the ribosome. And so these anticodons are also complementary to the codon, which means actually the anticodon is the same sequence as the DNA. So I'm going to explain that. If messenger RNA is complementary to DNA and transfer RNA is complementary to the messenger RNA, then these two are the same. They're equal. Kind of a math thing, but we'll also do an example so that you can see that. So the original DNA should be similar to these anticodons, except of course DNA doesn't use uracil. It uses thymine. So the original, did I do that right? I think I did. I'm not sure I did that right. I have to do another example. But nonetheless, this is the summary. We go from messenger RNA to amino acids, but transfer RNA is needed to bring the amino acids to the ribosome. So here's more of a pictorial viewpoint, with a cell. So we start with the nuclear DNA, and transcription occurs, and we make a primary transcript. The primary transcript is processed, and we get mature RNA because the introns are cut out. It's modified. Then the messenger RNA leaves through the nuclear pore, goes to a ribosome, and that's where translation takes place. So codons are essentially interpreted by the ribosome as amino acids, and we get a polypeptide synthesized. So this is the table that Watson and Crick developed when they cracked the genetic code. And they have written it not with transfer RNAs, not with DNA codes, but with messenger RNA codons. I'm sure it has to do with the order in which they discovered things, and we could backtrack and we could turn this table into DNA if we wanted to, but it's the messenger RNA that the transfer RNA actually looks at and brings the amino acids, so this makes sense. So the way you read this code is the first letter is in columns. The second letter, oh, I did that wrong, sorry. The first letter is in this column going down, so U, C, A, G. The second letter, you go across, U, C, A, G. And then the third letter, you go down in each of the rows, the big rows. You aren't going to have to do it that often. It's not like you have to remember that. You can just hunt and peck. It works. For example, if I just blindly said, find the amino acid that's coded for by mRNA CAC, you could probably find it somewhat quickly. Like I didn't know where it was and I just picked it out right here. That would put histine in the protein. Almost done. So what we're going to do in class together is we're going to start with the DNA sequence, write out a complementary mRNA sequence, and then use the table to predict an amino acid sequence for a protein. Then we'll probably backtrack. Not from here. We'll actually go this way. And we'll say, okay, well, what were the anticodons? If this was the messenger RNA, what were the anticodons? And then you'll look and you will see, let me get a different color out here and clear this all out. You will see that the DNA, TAC, is pretty equivalent to the transfer RNA anticodons, right? So CGC, right there, CCT, well, CCU because we don't have T. So you'll see that the transfer RNA anticodon sequence is really the same as the DNA, except you replace the T's with U's. So that's what we'll do together in our live class. Thank you.